Good afternoon, all. I'm representing Group 5. Our topic is immunity and vaccination. I'm going to deal with immunity. So, under immunity, I'm going to discuss.
Wait. Can you show your face? Where are you?
Eyes.
It's not possible? Okay, then continue.
Okay. So, under immunity, I'm going to discuss immune response to COVID-19, immunological spectrum, how long does immunity last after infection, and herd immunity. So, first, let's discuss about immune responses to SARS-CoV-2. There's two types of response, that is innate immune response and adaptive immune response. And this innate immune response is the first phase and the early response. So when virus enters the cell, it is recognized by the pattern recognition receptors. Actually, since this SARS-CoV-2 is a single-stranded RNA, here the receptors are toll-like receptors 7 and 8. And this pattern recognition receptors, after recognition of the antigen, they induces the adaptive proteins and which results in the production of interferon regulatory factor, NFKB, and activated protein 1. This further causes the activation of interferons 1 and 3 and different chemokines. These chemokines attracts the other innate immune cells like polymorphonuclear leukocytes, monocytes, NK cells, and these cells further produces chemokines like MIC, MIG, and MCP1. These chemokines recruit cell lymphocytes which recognizes viral antigens presented by the dendritic cells. Now, about the special characteristics of SARS-CoV-2, these virus in these infections, there is reduced interferon 1 and 3 responses. These viruses have better capacity to replicate within the pulmonary tissue, and there is significant induction of pro-inflammatory chemokines like interleukin 6 and tumor necrosis factor. And these SARS-CoV-2 elude from the antiviral effects of interferon 1 and 3. And they activate immune response and induce the production of cytokines, which is required for recruitment of the adaptive immune response, which is the phase 2 response. And adaptive immune response is a specific response to the infection. It involves two types of cells, that is T cells, which involves the cellular response, and B cells, which is the antibody response. Now about the T cells, there is two types of T cells, CD8 plus cytotoxic T cells and CD4 plus helper T cells. These cytotoxic T cells, as we all know, kills the infected cell, and helper cells stimulate the B cells to produce antibodies which are specific to the virus. And in case of SARS-CoV-2, there is 80% of the infiltrating cells involved are CD8 plus cytotoxic T cells. Now about how the CD8 plus T cells destructs the virus infected cells. Actually, as I mentioned earlier, this pattern recognition receptors recognizes the antigen, and the antigen presenting cells are presenting these antigen along with the MHC1 molecule complex to the CD8 plus T cells. So these T cells releases perforin and Gram signs, which causes the apoptosis of the infected cell. And before going to the antibody response, let me give a brief idea of the structure of COVID-19 SARS-CoV-2, which was already discussed. It has membrane glycoprotein and spike protein and envelope protein. And this spike protein is the most discussed one, and it is the most important. It has two parts, that is S1 and S2. And this S1 part is the bulb-like part, which has receptor binding domain, which is this receptor binding domain is for the ligand of the host cell membrane. And this stop part is involved in the fusion of this virion with the host cell. And now about the antibody response, that is this B cells produce the antibodies against this spike protein, and thus it blocks the virus attachment to the susceptible ACE2 ACE2 plus cells. This ACE2 receptors is responsible for the internalization of this SARS-CoV-2 into the cells. So as a result, if it is blocked, the invasion of the virus is affected. And during the antibody response, at first IgM and IgA antibodies are produced during the first week of after the onset of symptoms, and this IgA antibodies are fundamental to the mucosal defense. And next comes this IgG antibodies, which are detected at around 14 days after the initiation of the symptoms. And there's anti-S antibodies, which is bound to the IgG-Fc receptor 2 plus, enhances the viral entrance, and results in the production of pro-inflammatory cytokines. In case of dysfunctional response, unable to inhibit the viral replication and elimination of the infected cells, it results in an exacerbated inflammatory response, leading to cytokine storm manifested by severe acute respiratory distress syndrome and systemic consequences such as disseminated intravascular coagulation. This T cells, B cells, and macrophages, dendritic cells, etc., do not express this ACE2, but the dendritic cell is signed, acts as trans receptor for SARS-CoV-2, and it may transfer the infection to other susceptible cells. CD26, which is usually involved in T cell activation, may bind to the spike protein and results in non-productive T cell infection. And this may be related to the lymphopenia and activation-induced cell death. Now about the memory cells. Once the infection is over, T cells and B cells decline, but the memory cells will remain, and they respond rapidly if they come in contact with the same virus again, killing the virus and accelerating an antibody response. Now about the immunological spectrum of COVID-19. Despite of the high infectivity of the COVID-19, not everyone exposed to SARS-CoV-2 becomes infected. So there may be many reasons for that. It is possible that a small occasional inoculum does not reach a lower respiratory tract where susceptible target cells are seen, or there may be some unidentified genetic condition. But anyway, there must be a protective immune response in patients with asymptomatic or patients who are asymptomatic or with mild infections, and in those who have moderate infections which do not progress to severe form. This response must be capable of inhibiting viral replication and eliminating the infected cells with minimal tissue damage. Now, the effect of age, immunosenescence in elderly affect the T cell adaptive response, which is the phase 2 response in case of viral infections. And in case of treatment with ACE2 inhibitors in hypertensive adults, it results in the upregulation of the ACE2 expression and thus increases the susceptibility to SARS-CoV-2. And now, how long does the immunity last after COVID-19? We don't know for certain if people are immune to reinfection because not enough studies have been done. However, a study which was published in journal Immunity found that people who recovered from COVID-19 had memory B cells and memory T cells with hallmarks of functionality. And the antibodies produced during the infection is found to be maintained for at least 3 months after the infection, and it may go more than that to 5 months or 7 months. And this memory cells gives a quicker and stronger response the next time we encounter the invader. Thus, these cells, along with the antibodies, will likely to protect people from symptoms and further transmission. However, we cannot say that if there is a positive antibody test doesn't mean the person is immune to virus. It just means that the person has been exposed. And in some cases, these antibodies may provide protection. In some cases, they may not prevent the infection, but symptoms may be milder. And in other cases, they provide no protection at all. Now about the herd immunity. Herd immunity is the concept that if enough people are protected from infection, either by gaining immunity from having infection or receiving a vaccine, then the chance of a non-immune person contracting the disease is exceedingly low. But however, the infection protection by gaining immunity is nowadays not much preferred. And you can see in this diagram that if more person is immune, the disease transmission would be very less, and we can control the pandemic to an extent. And that's all. Thank you,
Mm,
ma'am.
it's over.
No, ma'am. The next topic is vaccination, which will be taken by.
Yes, yes. Please continue. Continue.
Good afternoon.
Yes. You can show your face and talk. The other person was not showing. Shafna,
Yes, ma'am.
you can open your video and talk. Yes. Okay.
As we learned about COVID-related immunity, and now it's high time to speak about corona vaccine. Myself, Shafna KM, here I am dealing with vaccines. As we know, a vaccine is a biological preparation that provides active acquired immunity to a particular infectious disease. Now, there is an urgent need for COVID-19 vaccine development. World Health Organization and its partners are committed to accelerating the development of a COVID-19 vaccine while maintaining the highest standards on safety. Before introducing a vaccine from lab to clinic, it has to undergo many testing processes. Preclinical testing, phase 1 safety trials, phase 2 expanded trials, phase 3 efficacy trials, early or limited approval, combined phases. One way to accelerate vaccine development is to combine the phases. And then post, if investigators observe worrying symptoms in volunteers, they can put a trial on pause. First one is preclinical testing. Scientists test new vaccines or cells, then give it to animals such as mice or monkeys. Phase 1 safety trials. Vaccines to small number of people to test safety and dosage as well as to confirm that it stimulates the immune system. Phase 2 expanded trials. Vaccine to hundreds of people split into groups to see vaccines act differently in them. Next, phase 3 efficacy trials. Vaccine to thousands of people to see how many become infected compared with volunteers who received a placebo. This trial is large enough to reveal evidence of relatively rare side effects. Next is about genetic vaccine. Vaccines that deliver one or more of coronavirus on genes into our cells to provoke an immune response. Here are some of the genetic vaccines. First one is Moderna phase 3. Develops vaccines based on mRNA to produce viral proteins in the body. The vaccine contains genetic instructions for building a coronavirus protein known as spike. When injected into cell, the vaccine causes them to make spike proteins, which then get released into the body and provoke response from immune system. Next one is Morningside phase 1 and 2. Imperial College London researchers have developed self-amplifying RNA vaccine which boosts production of a viral protein to stimulate the immune system. Gene-based vaccine carry genetic information for the host cells to make antigen which closely mimic natural infection. In case of coronavirus, the antigen of interest is a surface spike protein the virus uses to bind and fuse with human surface. Third one is Pfizer-BioNTech phase 2 and 3 combined vaccine. New York-based Pfizer and German company BioNTech made history based on preliminary data indicating that their coronavirus vaccine are 90% effective. They found that one version called BNT162b2 produced significant fewer side effects such as fever and fatigue, so that they included in phase 2 and 3 trials. Next is about viral vector vaccines. Vector acts to carry virus into humans. Some viral vector vaccines enter cells and cause them to make viral proteins, while other viral vectors slowly replicate, carrying coronavirus protein on their surface. First one is Johnson & Johnson phase 3. Beth Israel Deaconess Medical Center in Boston developed a method for developing vaccine out of a virus called adenovirus 26. The vaccine has provided protection in experiments on monkeys. Second one is AstraZeneca or University of Oxford phase 2 and 3 trials. Vaccine based on chimpanzee adenovirus. Vaccine raised antibodies against coronavirus as well as other immunodefense. Most viral vaccines are attenuated and inactivated viruses. Once inside the cells, the viral vector hack in same molecular system as SARS-CoV-2 and produce spike protein in 3 dimensions. It resembles natural infection triggering inflammations and mustering T and B cells. Human adenovirus, simian adenovirus, measles vector vaccines mimics a natural viral infection by forcing our bodies to produce viral proteins inside our cells that spurs the T cells of our immune system to attack these vaccinated cells and also seek to destroy cells infected with real virus in future. Now coming to limitations of viral vector vaccines. Human adenovirus circulate widely causing common cold, and some people harbor antibodies that will target vaccine making it ineffective. Use of attenuated chimpanzee virus in COVID-19 vaccine means it cannot now be used for malaria because those vaccinated for coronavirus have antibodies against the vector. Live vaccines of measles vaccine strain means that it could not be given to immunocompromised individuals.
Hello.
Ma'am, I couldn't change.
You couldn't change. Yes, it is coming.
Okay.
Next is about protein-based vaccines. Vaccine contain coronavirus proteins, but no genetic material. Some vaccine contain whole proteins, while some contain fragments of them. Some pack many of these molecules on nanoparticles. First one is Novavax phase 3. Maryland-based Novavax make vaccines by striking proteins on microscopic particles. Cuba's Public Health Ministry announced trials on a vaccine called Soberna. The Vector Institute, Russian Biological Research Center, state 1 and 2 trial for coronavirus vaccine they called EpiVac Corona. Second protein-based vaccine is Sanofi phase 1 and 2 combined vaccine, which is developed COVID-19 vaccine based on viral protein. They produce the protein with engineered virus that grow inside the insect cells. Next is about inactivated and attenuated vaccines. Vaccine created from weakened coronaviruses or coronavirus that have been killed with chemicals. Wuhan Institute of Biological Products developed an inactivated virus vaccine which the state-owned Chinese company Sinopharm put into trials. In collaboration with Indian Council of Medical Research and National Institute of Virology, the Indian Bharat Biotech designed a vaccine called Covaxin based on inactivated form of coronavirus. Studies on monkeys and hamsters found that it would provide protection against infection. About live attenuated vaccine. New York-based Codagenix developed vaccines based on live attenuated virus, but with a twist, they create viruses from scratch. Researchers rewrite the genome of viruses, introducing hundreds of mutations. Then they manufacture RNA molecules encoding the rewritten gene. Repurposed vaccine. Repurposed vaccine, which is vaccine already in use of other diseases that may also protect against COVID-19. Bacillus calmette-guerin BCG was developed in the early 90s as protection against tuberculosis. The Monash Children's Institute in Australia is conducting phase 3 trial called the BRACE to see if the vaccine partly protects against coronavirus. Summarizing the topic, scientists are racing to produce a safe and effective coronavirus vaccine by next year. 13 vaccines, including Pfizer, Moderna, and Oxford, reached the final stage of testing. Some of these will fail, while others may end without a clear result. But few may succeed in stimulating the immune system to produce effective antibodies work against virus. We have collected these informations from NY Time Vaccine Tracker site, PubMed, and journals. There is too much information about corona vaccine and its trial, but because of this limited time, I mentioned some of them only. Before concluding, I would like to specially thank Dr. Alice ma'am and our group members for their help and support. Thank you. Thank you, everyone.